philosophic philosophy event, but yeah. So I am the omega before the panel discussion. Um, I'll simply continue. Yeah, yeah. Okay. And well, if you're the last in such an event, and I didn't have any abstracts or so, I just had titles, so I had to somehow guess what the others say. So I'm not too rapid presentation right now because it gave me the last bits and pieces that I need for my presentation. So why? I mean, Mark said we cannot be. I'm a statistician by training. I sometimes have a bad conscience when I call myself a statistician because I haven't been doing statistical analysis for a long time now. I do rant about papers that use bad statistics or bar charts or something like that, or when they use standard errors of the mean for their whiskers. But yes, it's true. I mean, if you just felt a little bit lost during Felix's presentation, that's because he's a bioinformatician. Why do we have bioinformaticians? Well, next generation sequencing. That's caused it. You know, suddenly your average, I mean, suddenly biology got interesting again, but the problem was you cannot analyze 100 whole genomes on your laptop computer anymore. Suddenly you need command line tools. What are command line tools, you know? And a bit over 9 years ago, I started directing this core facility Nexus. And I hired bioinformaticians. I didn't even know what they exactly are doing, you know, but I had to hire them because we had to handle NGS data. And I learned that, interestingly, a lot of, or let's not get ahead of ourselves. Let's go to the first lady. Okay, so this morning, Valentin told us the definitions. I am really happy that he chose the definition of reproducibility, of the different types of reproducibilities that I am also most fond of, which is the one by Goodman. And I basically, yesterday, I saw for the first time a publication title that has a p larger or smaller equal 0.05. And today I saw 4 different publications having p's in their titles. Okay, so I can save the definitions of reproducibility. Then Romain spoke about transparency. So how to report, because the reporting part is also part of reproducibility. And we heard also Valentin say it's more the communication actually. So how do we report things? And then we learned about the Holy Grail, I mean, systematic reviews or the actual meta research that gives us the answers. It is at the top of the pyramid of evidence. So yeah, but what am I doing here? Now I try to shape this thing called computational reproducibility. So what does it actually mean? Huh? I'm somehow convinced, or I'm trying to shape the concept of computational reproducibility towards the actual way to get to reproducible results, conclusions, inference maybe. And we do that by having 2 things. Let me bring in the tools. So Felix now showed us a lot of nice tools, which we use on a daily basis in my team as well. So the question about which workflow management system do you use, Nextflow or SnakeMake, that is discussed on a daily basis in our coffee breaks, you know. Our conclusion now is we go hybrid. We do both. We use SnakeMake for a couple of things, and we use Nextflow for a couple of other things. You maybe not have seen it, but when he showed the screenshot, he was logged into the NFCore website. NFCore is a specialized set of pipelines that are meant for core facilities, and I run a core facility. So when we have fairly standardized data analysis, we tend to simply use the off-the-shelf pipelines that we have from NFCore, so they're nice. We started using SnakeMake, so there is sort of like an a remainder of SNAKE in there. So I believe if we have tools, what we feed into these tools also needs to be proper. I mean, you know, the SISO problem, the data needs to be fair. And I actually expected that already somebody told what fair is, but I assume that let's show a show of hands. Who can name the 4 letters of fair, what they mean? Oh, it's on the slide. Who would have been able to do it without the slide? No? Okay. So basically, these are attributes for data. And they're in high, there's a little, it's a sort of a little bit of buzz around this whole concept, but it is indeed super important. It's also important when we want to go down the road towards like artificial intelligence and all these things, you know? I mean, even the best artificial intelligence will not be able to train its regression models. I mean, that's what they are, if it doesn't have the proper data to do so. So the data needs to be findable. It needs to be accessible, A. It needs to be interoperable. That means standards, naming the same variables in the same way and having the same levels and so on and so forth. And reusable, that's sort of the conclusion of the first 3, and each one of those is important. Because if the data is then fair, we can start using the right tools. So tools, and I will quickly make a schematic of, when I think about the research project, that's what comes to my head, you know. So of course there is an experiment before we get to raw data. And I also like to speak about how we can do the experimental side of the lab more reproducible. And we can also do that by scripting. So if we use lab automation, for instance. But let's start at the raw data. So we process the raw data in some way. For instance, if I use the bioinformatics example of DNA sequencing, we need to take the FASTQ files that contains the reads. We need to align these reads, and we end up with some enriched data. Let's call them a variant calling file or the genome of interest that we have. Then we have an analysis that gives us results and we want to publish. So if we look at tools, I mean, for raw data and or enriched data, there are open repositories like OSF or the Open Data Repository. There is also, I mean, you cannot put, or you should not put a lot of data into version control. Here you can see the 2 typical tools or languages that we use when we are working in my group, which is Python and R. And for these, you have these notebook environments. So the Jupyter Notebook is understand it like a website that allows you to actually combine code and text with each other. The same is true for the R Markdown. They have the R notebooks that you can have in RStudio that allow you to combine, it's sort of like the literate programming. You can add code and text together. The summit of this is that your whole publication is basically a code, and you could have your whole publication, all the figures and all the tables in the publication reproducible from code if you add the data, the experimental data that you need. So then here are the workflow managers. So they sort of span the processing and the analysis up to the results. They could even go further. The arrow could extend further out. But mostly what we do is we steer the different steps in the processing and in the analysis, because we sometimes use different tools for different steps. So there is, for instance, a tool that helps us take reads, raw reads, and align them. And then there is a tool that is actually really good at taking the aligned reads and finding the different variants in the genome. But these tools may have different versions. They may need different configurations. And that's what the workflow managers do. You can tell them when to use which tool with which input. Then version control, Git. So these are the true platforms which are most well known, I would say. So GitLab and GitHub that use the version control Git that allows us to go back in time and see what we did once. We have to document which steps we do it. Usually, or a lot of people say that Git is super important when you work in teams. I would say that a team size of 1 is sufficient to use Git. Then when we are doing the analysis, generate results, and maybe even go to a publication scale, then there are also platforms like Zenodo or F1000 Research that allow you to have actually Markdown, or for the enthusiasts, LaTeX scripts to sieve them into a finished report. I just saw in the break that there are even executable research papers now on eLife that basically also ensure that the code, the analysis aspect, and what is put into the paper is actually in one go reproducible. And then, of course, the containers are also here. So they ensure the actual problems of which we often face when we look at what is also below our programming language. So we run Windows, we run a Mac OS, we have maybe version 3.5.17 of our studio and not 3.7.18. And containers ensure that these problems don't happen. That basically means if something is missing, it should be in the container, so I can take the container to another researcher. They simply run the container and all the software is installed inside the container. So it seems like the computational guys got it all figured out. And yes, they have the tools, but that's not the only thing that is important here. So maybe some of you have heard that nowadays you can basically draw a little bit of blood, check what your microbiome is, or I don't know, maybe it's a stool sample, check what your microbiome is, and then predict in which stage cancer you are. So this publication here by Poor et al., they claimed this. It was a highly cited Nature publication, and there has been downstream research made on it. I think some 11 or 12 paper that based on their signatures, a company was founded, a startup was founded. Well, the problem is that 3 years, no, a couple of months later, actually, the guys around Steven Salzberg tried to replicate this. They were also working on microbiome signatures in cancer. So this is not that they are in spite doing that. I mean, if your research, let's say, peers are doing something interesting, of course, you'd go there and you check what can we salvage from this and use for our own research. So they went there, they tried to reproduce it, and they found 2 problems with the study. The first problem was they, whenever you use a database with pathogen genomes, you need to filter out as much as possible human DNA, because a lot of unfinished genomes, so let's call them like version 0.9 for this pathogen, they are filled up with human DNA. So instead of finding pathogen DNA, they were finding a lot of human DNA because they didn't filter hard enough. And the second problem they had somewhere in their code, they made a mistake, and they let the machine learning algorithm see the labels of the different cancer types. So it generated, due to that, it generated a fake signal that basically associated cancer types with some of the microbes. They found this out because they saw that for some colorectal cancer, a certain bacterium that lives on corals in the sea was indicative, and that's not appearing in humans, you know. So the tools worked in the end, because the data was available. Nowadays, it's really hard to actually publish omics data without publishing the data. And the description of the process, so there was no code published with the study, but they were able to actually find out what went wrong. Rob Knight then sort of said, yeah, well, it's just a preprint. I'm not going through 17 pages of debunking. And we did another study that proved the same results. So maybe going into the direction of inferential reproducibility, maybe. However, the paper that I showed you before, the debunking paper, that's now fully published. And a couple of weeks later, this happened on Nature. So actually they put a flag there. It's not a red flag, but it's a yellow flag that basically warns you, hey, there is something weird going on. When I then read the Givadi paper, this is on their GitHub page, you know, it says, for those interested in the code, all of our methods are based on free open source software. We ran our labs, blah, 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 blah. So it should be easy for others to replicate our results. I wouldn't have been much asked to give us a bash script or maybe a config file for Nextflow to reproduce it. But tools are not the only thing. So the fair data can be processed by the tools, and the tools help us do that. And all these tools, reproducibility is not evenly distributed across fields. Software developers, they're totally reproducible all the time, you know? So having the computer sciences flow into biology with bioinformatics, and then now we get a lot of biostatistics, and everything is turning about health and life sciences anyway. So this is why we now suddenly have this, we need to learn these new words, you know, like containers and stuff like that. But it goes further, you know. So what we need is a culture of sharing and openness. This means we need to get rid of being afraid to be scooped, or being afraid to do the extra mile of becoming reproducible, or at least put up the extra mile to share your data in a platform like the European Genome Archive. We need to incentivize reproducibility. So let's say we have guidelines and we have policies that actually, well, it's maybe not incentivizing, it's more like motivating people. And we can have awards and rewards for people who are reproducible. And then education and training. So everybody should simply get their computational skills up. Everybody needs to be able to code at least a little, or at least understand where to get the proper help to do so. And then also teach how reproducibility awards you as an individual and your research when you actually have these open and reproducible practices. Many of you were nodding. I think that a lot of what I just said is BS, by the way, because that's just more or less what you get when you. I also asked ChatGPT a couple of questions. So I asked him, what should we do to be reproducible? And it spat out a couple of these points here. Scooping? Scooping? Come on. Who's afraid of scooping? That's the worst excuse for not sharing your data and code and being like a dinosaur when it comes to data analysis. I mean, when do you share your data and code? When you're publishing. What's this scooping idea? It's terrible. I mean, sending out your publication, your manuscripts for review is more dangerous than basically putting it on a preprint server that has a date stamp on it. Cannot be scooped on that. The extra mile for becoming reproducible, the downstream benefit from having proper scripted science is obvious. Is it really true that your supervisor is forcing you to click through GraphPad Prism every time? Are you kidding me? And aren't they not realizing that when they say, I want you to redo this figure with this and this and that, when you come after 30 seconds with the new printout, because certainly they want it printed on paper, they realize, oh, that was fast. How did you do that? Code. Yes, yes. but not guidelines. Let's make it really rules, huh? Let's make it regulations, that people need to share their stuff, that people need to put their code online. Nowadays, in omics publication, it's all, at least in the health field, it's almost impossible to make claims about gene expression data or so if you're not sharing the data. That's how it needs to be. It starts at the journals, but maybe it must not end there. Maybe it has to go further than that. And I'm speaking also about, consequences when you receive funding. When you receive funding from the public hand, and there's guidelines in the call about how you share your data and your code, and you don't do it, you need to be reprimanded. If your expedition to the Antarctica is financed, and you drill your ice cores out there, and then you don't share your data within 6 months, you will not get funding. Afterwards, I mean. So consequences, not only regulations, but consequences. Awards, come on, we're in Switzerland. We're the most egalitarian country in the world. I mean, our skiing athletes keep their medals in the basement. So awards, yes, it's nice for a PhD student or so. But this cannot be understated. I mean, get your training. Don't be so lazy. Learn computational skills. At least have a concept of them, and then hire a colleague that speaks your statistics language or whatever language you speak, and basically can talk with you. So we saw it, that not one person can do all these things. We saw that in Davos, they mix people of different flavors to actually achieve good cardio care. And that's how we do it. I have 5 different profiles in my team, and they work basically on each project together, somewhere along the whole chain of events. And yeah, this training of what reproducibility is good for and stuff like that. I mean, this is supposed to be a watch. Well, time is working for us. Someone who does not want me to name him here because I'm going to cite him on something said, well, science progresses funeral by funeral. And I mean, this is so true. When I teach statistics to to the medical students, they always come with their, I always tell them how bar charts are stupid and how you need to do a sample size estimation. And I tell them what p-hacking is and harking. And then they come to say, yeah, my supervisor told me this and this and that. I said, yeah, but have you told him what I told you? Yes. They simply said, they want to do it like they want to do it, like they always did. So they will go. With time, they will go. Thank you very much. I'm also referring here to our first National Reproducibility Conference in Zurich. Please come, and I'm happy to take your questions.